Hello, we are Cheyenne, Henry, Adriana Richardson, and Maya Blossoming, and today we are presenting on the effect of single nucleotide polymorphisms on mismatch repair mechanism and the MSH2 gene.
So HNPCC is an inherited cancer that occurs due to mutations in the mismatch repair genes. Due to this, cells lose the ability to repair mismatches in double-stranded DNA, which can cause tumors and increase the risk of multiple types of cancer. So MMR is a system for recognizing, repairing insertion, deletion, and misincorporation of bases that can occur in DNA replication and recombination, for an example. So MSH2 is a tumor suppressor gene that codes for the DNA mismatch repair protein. Thus, a mutated MSH2 gene can increase the chance of developing certain cancers. So overall, HNPCC is impacted by the presence of mutations in MSH2, meaning that if there are mutations in MSH2, that can decrease the functionality of the MSH2 repair mechanism, which would increase the likelihood of the development of HNPCC.
SNPs or single nucleotide polymorphisms are a type of polymorphism that involve the variation of a single base pair. SNPs are used in human genome to study how they correlate with disease, drug response, and other phenotypes. Sometimes a SNP does not change the protein sequence, thus tying into our hypothesis for this project that the SNP in the MSH2 gene that is affecting the plasmid will affect MSH2, which would then decrease the efficacy of MMR and thus lead to high mutation rates. In order to test our hypothesis, we used yeast to model the mutation rate to observe the impact of our group SNP on the MMR mechanism.
So the purpose was to extract plasmid DNA from E. coli strands to use these plasmid DNA samples to identify PMSH2 and determine the mutation rate for each plasmid sample. We extracted plasmids from 3 E. coli strands, SCX, SCY, and SCZ. Overall, E. coli has the same replication origin and it's a similar model mechanism, which is why it is used. And plasmid provides this in order to allow the E. coli to survive and proliferate in the presence of ampicillin, AMPR.
The objective of this lab experiment was to learn how to use a polymerase chain reaction to amplify a portion of the MSH2 gene in order to locate the mutation and distinguish the plasmid MSH2 wild type from the mutant plasmid MSH2. APE was used to identify plasmid's template strands and the 2 primers, i.e. the forward and the reverse. The polymerase chain reaction was used to amplify specific sequences of the plasmid to digest the samples later. From this information, the restriction enzymes were located and used to cut the specific sequence of the MSH2 and digest the samples to find the mutations present on the strand. After the PCR samples were amplified and digested, gel electrophoresis was used to determine the number of fragments and the size of each fragment in order to identify the wild type or mutant plasmid MSH2.
So the next part of this lab was to use yeast transformation. This was done to transform the yeast strain AGY75 with the plasmid samples that were differentiated from before, in this case, the mutated plasmid. Furthermore, this lab aimed to determine in the specific environmental conditions would the yeast grow in the presence or absence of a plasmid. In order to do this, the yeast were grown on a specific environment, which was the SD plate, which had minus histidine, minus tryptophan, and minus uracil. This method in particular compared the growth of the yeast with and without the inserts of plasmid in order to eventually determine the mutation rate. Furthermore, yeast were used because of their ability as a good model to measure mutation rate. Finally, the PSH44, which was inserted with the PMSH2 plasmid in all 3 strains, allowed the yeast to grow in the presence of tryptophan and uracil. However, we were trying to see what's going to happen because of the histidine. The final lab that we did in the conclusion of this project was the FOA assay. The goal of the FOA assay lab was to measure the mutation rate in AGY75 strains with the different plasmids inserted. This is extremely important because in the presence of a functional ODC protein and FOA, a toxic environment is created which will thus kill the yeast. In the presence of a non-functional ODC protein, because of mutations that aren't fixed, no toxic environment will occur and thus there will be growth of the yeast. This lab was done by using a control group, the SD plate, which had no histidine, tryptophan, and uracil, and the other plate, which was the FOA plate, which added FOA to the environment. Each plate had 3 concentrations, OD1, 0.1, and 0.01, and the 3 plasmid samples were suspended on the plates and spread across the different plates, and the mutation rates were then determined for the wild type PMSH2, mutated type, and PRS413. Finally, the SD plate was used as a control group because we know what the growth patterns would be.
We found that in the 260 and 280 ratio, the values were 1 1.87, 1.82, and 1.90 for samples A, B, and C. These values are important because if the ratio is around 1.8, it's considered pure and not contaminated by other substances in the cell, like RNA, proteins, and salts. So this lab was important because efficient DNA plasmid DNA is important in extracting target DNA needed for later processes. So this is showing the gel electrophoresis that was used to separate the DNA fragments based on the size and charge in order to determine which plasmid DNA sample had the MSH2 insert. Lanes 2 to 4 were restricted enzymes to digest the plasmids, and lanes 5 to 7 were undigested. So after digesting our plasmids, it was found that sample A, which contained one fragment, was the PRS413 because it had no restriction sites for MSH2, while samples B and C represent the mutated and wild-type PMSH2. It's found that in sample A, B, and C, the estimated size was 5,000 kb, and this is important because it allowed us to differentiate between the inserts with the plasmid and without the plasmid.
The wild-type and mutant plasmid MSH2 samples were digested and cut using the restriction enzyme BSM-AI in order to determine the number of fragments and sizes of the mutant and wild-type MSH2. It is found that sample A was the PRS413, and sample B was the wild-type MSH2, and lastly, sample C was the mutant PMSH2. Sample A was the PRS413 because there was no PCR product amplified or digested. This was reasonable because the PRS413 contains no insert of MSH2 and thus would not be recognized nor amplified during the PCR process, hence creating no fragments during digestion. The wild-type and mutant PMSH2 samples were distinguished because the restriction enzyme recognized two sites on the mutant PMSH2, creating two fragments, but only one site on the wild-type PMSH2. Additionally, it was found that our mutant 4, which was on the wild-type PMSH2, had a change of glycine present at the 704th nucleotide base, which was then mutated to the aspartic acid. This will be useful in the future to use with the FOA assay to determine what will happen to the mutation rate when there is a SNP on the MSH2 plasmid insert. Here's more of our experimental data highlighting the fragments and estimated size of the fragments of the plasmid samples.
The next part of this lab was the yeast transformation. As I mentioned, yeast are good models to measure the mutation rate and have similar replication origins to the plasmid. The results of this lab were important to determine if the yeast would grow in the presence or absence of a plasmid, with the presence or absence of a plasmid in the specific environments. In this case, the environment was the SD plate with no histidine era. So sorry, or tryptophan. The PSH44. I'm so sorry. So sorry, y'all. I'm so sorry. Wait, hold on. I'm so sorry I missed this recording. The next part of this lab was the yeast transformation lab. As I mentioned, yeast are good models to measure the mutation rate and have similar replication origins to the plasmid. The results of this lab are important to determine if the yeast would grow with the presence or absence of a plasmid in the specific environments. In this case, the environment was the SD plate with no histamine, uracil, or tryptophan. However, the PSH44 insert provided both the tryptophan and ura to the cells. It was found that cells that incorporated the plasmid, PMSH2, PRS413, and PMSH2 wild type, mutant type, all grew in the yeast stream with no plasmid did not grow because it couldn't provide histidine to the environment. Furthermore, it was found that PMSH2 mutant type had 7.82 times 10 to the 2 colonies, while the sample with no plasmid had 0 colonies at all, which meant that only the plasmid insert would grow. The last part of our lab was the FOA assay lab. As I mentioned, FOA assay is extremely important to determine and compare the mutation rates of the different strains that we are studying. The mismatch repair mechanism is essential for this stage because PSH44 is prone to mutations because of the GT16.5 ura gene, and those frameshift mutations must be fixed. Before determining the results, we knew that what the growth pattern would occur for PMSH2 wild type and PRS413. For wild-type PMSH2, because it has a fully intact MSH2 gene, it will readily fix those frameshift mutations. Because of this, it will create a functional ODC protein. In the presence of FOA, will have a toxic and thus kill the yeast, as shown on the images 1, 0, and 0 growth. PRS413, on the other hand, has no MSH2 insert, which will be unable to fix those frameshift mutations and will create a non-functional ODC protein, thus we see growth 5, 2, and 0. Finally, we were trying to test what would happen to our wild type, or mutant type PMSH2, and it was found that it had a growth of 9, 0. This means that it acted closer to the PRS413, which tells us that the PMSH2 mutant actually had a mutation or a single nucleotide polymorphism that did impair the mismatch repair mechanism, making it unable to fix those frame shift mutations, thus allowing the yeast to grow because of that non-functional ODC protein.
So overall, our hypothesis for this project was that the SNP in the MSH2 gene that is affecting the plasmid will affect MSH2, which would then decrease the efficacy of MMR and thus lead to high mutation rates. Through our experiment, it was able to be determined in tandem with reference to the GAMI paper that the mutant study was MSH2, G693D. The human equivalent was determined to be G674S. Since the mutation was found to affect ATPase activity of MSH2, that means that it completely abolishes MMR functionality, meaning that the MMR mechanism is lost, which is fully supported by the experimental data as explained by Maya.
Mutations in the MSH2 gene are detrimental to mismatch repair mechanism, which in turn leads to higher risk of developing cancer, HNPCC. This project was important in studying the way SNPs and mutations potentially affect the mismatch repair mechanism. We can conclude that SNPs decrease the efficiency of MMR, leading to harmful mutations, and in the future, understanding SNPs can play an essential role in developing preventative treatments to target and decrease the risk of cancer and tumor development.
These are our references for our presentation, questions, and thank you. Special thanks to Dr. Gosia and Dr. Mojib.